Let's get started here. Our first speaker this afternoon is Dr. Justin Randall. And Dr. Randall's lab is here at UMKC, and they are finding new treatments for AMR infections, which are now among the leading causes of death worldwide. They're trying to discover new potential peptide antibiotics and determine the mechanism of action, structure, potency, and toxicity. I want to say those again. Action, structure, potency, and toxicity. Now, I repeated those because these are also four considerations Justin has when he determines what the perfect homebrew beer is. Justin's a home brewer, so he's interested in structure and potency and hopefully not toxicity. But while the beer's fermenting, he's either playing keeper on a 5-on-5 soccer team or bowling on a league. So throw an ace. Welcome.
All right, great. Thank you again for the invite. I'm new to UMKC. I'm new to Missouri. I've only been here since August, so I'm really excited to be here and tell you all about a little bit of what we're doing in my lab. It's just started up. We're just getting some research going. It may seem kind of weird that I'm starting off with a sand dune as a picture on an antimicrobial peptide talk, but hopefully that'll make more sense in just a second. If you're playing my Where's Waldo game, the peptide was hiding back in the shadow of that sand dune. And this is my favorite antimicrobial peptide. It's called Protegrin 1. If you're not familiar with peptides, they're just strings of amino acids, small proteins basically. Protegrin 1 has 18 amino acids. It's cationically charged and it folds into a beta hairpin structure. I just want to make everybody realize why I love peptides so much. The diversity of sequence for a short peptide is astronomical, and that's a truly astronomical number. If you take just an 18-length amino acid peptide like Protegrin 1, and you let each of the 18 positions be any of the 20 possible natural amino acids, you end up with 1 times 10 to the 25th total sequence diversity. That's actually 10 times more than there are estimated stars in the universe. So within that pool of peptides, I truly believe that you can find one that can do just about anything that you want. And my interest is finding things that kill bacteria and other pathogens while not harming human cells. So amongst that big pool of peptides, so far we found about 3,000 known antimicrobial peptides. Problem is they're not great therapeutics because they are also toxic. They're potent killers of bacteria, fungi, even viruses, but when we try to use them as a systemic therapeutic, they often have toxic side effects which don't allow them to be used as drugs. So what I'm actually interested in doing, and what this talk will mostly focus on, is how do we adjust Protegrin 1, its sequence? How do we find the actual grains of sand in this sand dune that are variants of Protegrin 1 that maintain their antibacterial activity, but they lose the toxicity effects, so making it a better therapeutic. How do we sift through that sand is really what this talk is about. So just an overview of my lab. We're interested in finding new treatments for antimicrobial resistant infections, as I said. That's mostly because we have this real big problem, which I'll get into in a couple more slides, where antimicrobial resistant infections are causing a lot of deaths around the world globally right now, and it's only estimated to more than octuple over the next 30 years. And it's going to become, actually, it's predicted to become as equivalent to cancer deaths globally. So this is something that we need to work on now, basically. We don't have enough therapeutics in the pipeline to fight these antimicrobial resistant infections. And within this big bubble of finding new antimicrobials, our lab is really interested in peptide therapeutics, as I mentioned. And we want to target essential processes in the cell envelope of bacteria. That's basically the cell surface in between the outer membrane and the inner membrane. We target this area because it's unique to bacteria. Right? Eukaryotic cells don't have it, human cells don't have it. So if we can target processes in this region, we're more likely to isolate things that won't be toxic to human cells. And then within that, I'm really interested in membrane disruptive peptides. So peptides that actually target the membrane directly. And that's what Protegrin 1 is. And they usually target the membrane by either creating pores in it by self-associating with each other, or they act kind of like a soap or a detergent and just dissolve that membrane away. So with that, I'm just going to re-emphasize how badly we need new antibiotics. So this is a brief history of antibiotics in general. It starts at the very beginning in the pre-antibiotic era where bacterial infections are actually causing more death than any other cause of disease in the world. These can be gastrointestinal diseases like cholera or Clostridium difficile, or pneumonial infections that are infecting the lungs. We have no real way of treating them, so it's up to your immune system to kind of come up with a way to kill them. Then penicillin kind of revolutionized everything. All of a sudden, most of these things were treatable, but resistance got gained to penicillin very quickly, as it does to all antibiotics. So we needed more than just penicillin, right? And we hit this golden age of antibiotic discovery, which lasted about 30 years, where we found basically every antibiotic that we still use today, at least the classes of antibiotics that we still use today. And instead of continuing to find more, because that got harder and harder, we just started tweaking the structure of ones that we already had to overcome the resistance. The problem is, if you take something like penicillin and you adjust it into something like amoxicillin, it overcomes the resistance for a brief period of time, but that maintenance doesn't take a very long time for the bacteria to then gain resistance again to that antibiotic. So we really need new targets of antibiotics, not just new antibiotics in general. So that tweaking era really only delivered one new class of antibiotics, and since then there's only been one or two more all the way up to today. So it's already become a global health crisis across the world, and it's, as I mentioned, already a leading cause of death again globally, and that graph again is really scary in the future if we don't start implementing things now. And one of the really major problems that I want to emphasize is that these are not very profitable drugs to make. So pharmaceutical companies don't want to make antibiotics. You take them, you cure your infection after you take one course of them, and then they last about 10 years before resistance gets developed to them, and then they're no longer really marketable at all. So where's the profitability there? If you have to put in all the research and development to find that molecule, then you only sell it for 10 years, and you only take it once and it cures your infection, it's not really going to make you a ton of money. And that leaves academia as kind of the research and development arm of the pharmaceutical companies, and that's kind of where I feel like my lab fits in. Okay, so we need new methods of discovery. I think I got y'all there. I'm just going to give you a real quick story with a very limited amount of data. One more emphasis on why I like peptides so much. Small molecules are great, but they have a very low target binding diversity. So if you want to inhibit an essential enzyme, a small molecule is great at binding a pocket in that enzyme and inhibiting the activity of it. It's not so great at interrupting a protein-protein interaction that has multiple face of interactions with each other. So instead of using small molecules, I really think that biologics or peptide-based drugs are kind of the new revolution in looking for new drugs to cure many different diseases, not just antimicrobial infections. These include antibodies and proteins, or what I like to call the Goldilocks product is these peptides, and specifically for me, those are antimicrobial peptides, because they actually have a much larger target binding diversity, but they're not so large that they can't still access targets that are inside the cell. So back to the sand dune and the quick story that I wanted to tell you all about. We have a technique that we developed called deep mutational surface localized antimicrobial display, which is a mouthful, so we nicknamed it DM-SLAY, which is a lot cooler. The sand dune, again, we're trying to sift through all those different peptides to find the ones we're most interested in, right? But we're talking about billions of trillions of possible peptides, right? How do we begin to get that throughput? So we developed a technique where we engineer the bacteria to both create the drug, the peptide, and then put it on its cell surface and ask the bacteria itself to test its activity. So it's both the producer and the screener of the drug. And we do this just by incorporating DNA into the bacteria that encodes this system of surface display. And we can make these libraries as many as 5 or as many as 1 million different unique peptides that can be produced by the bacteria and then displayed on the cell surface. If you induce a peptide that has antimicrobial activity, it will kill that cell. So you actually get cell death for anything that's antimicrobial, but things that aren't antimicrobial will just continue to grow at a normal rate. So if we look at a culture of bacteria where we can have billions of individual cells growing in that culture, each cell can be its own experiment, where each cell puts a different peptide on its surface and then evaluates whether that peptide kills the cell producing it. So we can actually screen many different peptides just in one experiment. And we basically can evaluate this or quantitate this by looking at the reads on the plasmids that were put into the bacteria. So if we do next generation sequencing, we can quantify those reads and then compare induced to uninduced, and we expect that we get a depletion in antimicrobial peptides in the induced relative to the uninduced population. This is the simplicity of the screen. It's really, really cool. It also gives us information when we plot this chart induced versus uninduced, where we get inactive and active peptides. So we not only learn what's good at killing bacteria, but also what's bad at killing bacteria. So now that I set this graph up, I'll give you a little bit of real data. This was one screen for protogrin. These are 4,000 unique peptides that we evaluated the antimicrobial activity for, and we pulled 50 from each of these groups. So 50 peptides that were predicted active, 50 that were predicted inactive, and we tested them in vitro to see whether or not we were correct in our predictions. 86% of the time we were correct in those predictions, which was pretty great. We then took those 50 peptides and we recorded data on their antibacterial potency, their toxicity, and their membrane specificity. I don't have time to go into the details, but we use machine learning to train these different algorithms predicting these different features and use those to screen 5.7 million variants of protogrin 1. So now we're talking about a lot more sand than just that 4,000, right? We ended up finding 95,000 that were more bacterially specific. 75% of those were indeed more bacterially specific when we pulled them out and tested them in vitro. And I'll only tell you about our favorite candidate. It was BSPG 1.2. It's here in purple. It actually is twice as active against a multi-drug resistant strain of Acinetobacter baumannii, which has no capable treatments in clinic right now. And when we put it into a mouse, it's actually 10 times less toxic. So the maximum tolerated dose is 10 times higher in the mouse than the original sequence. So this is one way that we can begin to sift through a bunch of that sand. And then if you look at it in vitro, we see the same thing. Or sorry, in vivo. If we look at the just number of bacteria that are in different organs, we see that we actually get a much higher reduction when we treat with BSPG1 versus when we don't. So this thing works in vivo as well. So just back to the sand dune to recap, we got a lot more sand to go through. That 5.7 million is about a cup of sand. We can go through the entire sand dune for protogrin 1, and there's 3,000 more dunes for us to start digging through to find new antibiotics. And that's where I'd like to just leave it for you to think about. So I'll thank people who have been involved in this research, including my former mentor, people in my lab right now, which is small but growing, and then funders and the machine learning collaborators that we talked about. Thanks.
All right, thank you. Good job. Love the sand dune illustration, that was great. Questions? Yeah, please.
Great work. I love peptides as well.
Thanks.
I was wondering, in mouse models, are those peptides, how are they introduced?
Yeah, I didn't have time to go into it in detail. We did intraperitoneal infections, so we dosed the bacteria into the body cavity, waited a little bit, and then again dosed with either peptide or a mock treatment. Then we let them grow for, I think, 6 to 10 hours. I don't remember which one that data set was for. And then basically dissected them, took their organs out, grinded them up, and looked at the bacterial counts in each of those organs.
Yeah, because usually the issue is maybe the proteolysis of the peptides, But
Yeah, so we did have to put it kind of right where it needed to be. We tried to do a thigh model of infection and it didn't work. But that's not surprising because it's still a natural sequence peptide. So actually one of the things that my lab is doing now is making synthetic modifications to that peptide to try to make it less susceptible to protease and actually make it hopefully stay in the bloodstream longer and be able to find that target.
yeah, great.
Mm-hmm.
All right, don't forget the Mentimeter capability for your questions through the QR code on the screen. We don't have any questions there, so this is your chance, or the microphone is your chance. Ah, here we go.
Wasn't planning on asking a question, but then I thought of one, so.
You're kind.
Yeah.
Way to go.
So my question is, what have you found that it makes things susceptible to bacteria, but then not susceptible to human cells?
Yeah.
So what features of the peptide?
That's a great question. Again, I didn't have time to go into that in detail, but what we found actually is there's 2 disulfide bonds that form in protogrin 1 that help it wrap into that beta hairpin structure. We found that if we removed 2 amino acids involved in that disulfide bond, so the cysteines that were actually involved in that, and made them into more hydrophilic amino acids, that took away the human toxicity. Vice versa, if we added aromatic amino acids, especially tryptophan, anywhere into the sequence, toxicity skyrocketed for that particular peptide. So we're really interested in seeing whether or not that same trend is true of other antimicrobial peptides, or is this specific to just protogrin 1 and you kind of have to do each peptide individually? Yeah.
All right. We do one more if you've got one.
Yeah, I was just wondering if the timeline for the bacteria to develop resistance to these peptides you would expect to be similar to the existing drugs.
Yeah, another great question. One of the reasons, again, I love peptides so much is that especially these membrane-targeting peptides like protegrin 1, they don't have resistance developed to them very quickly. That's not to say it can't happen, but I personally in the lab have not been able to evolve a bacteria that has resistance to protegrin 1 because you're actually targeting the membrane itself. It's very difficult for the membrane to actually change its entire membrane dynamics. Whereas with a small molecule, all you need to do is change a couple of amino acids in the binding pocket of where that small molecule is, and you gain resistance to it. You might need whole rearrangements of the membrane in order to actually gain resistance to protegrin 1. So resistance is harder to develop for peptide-based therapeutics for that reason, but not necessarily impossible. We do see it happen in clinical strains.
All right, let's thank Justin.